let me welcome everybody to the EPS 75th anniversary meeting from wherever you're joining us. Thank you for taking the time. So the first meeting of the Experimental Psychology Group, it was called, took place in 1946, and Oliver Zangwill invited 8 people to join him in the formation of this group in Bartlett's rooms in St. John's College. I think their idea right from the beginning was that the EPS would especially focus on psychology as a scientific discipline and leaving the more professional side of psychology to the BPS. And obviously that's turned out to be rather prescient, that psychology would indeed become a major discipline of science. From the original 8 members, I think the EPS now has something in excess of 1,100. And if you look at the themes of that early work in the late 1940s, 1950s, they're remarkably modern seeming, in particular the emphasis on the mind as a processor of information, on decision making in an uncertain world, and on the possibility of integrating understanding from behavioral and neural levels. 1950s obviously were the time when information theory was first brought into psychology, 10 years of very busy work culminating in, for at least the British audience, in broad-bench perception and communication. 1949, Hebb's The Organisation of Behaviour, and the idea of the cell assembly introduced to the community. The 1940s, the Pitts-McCulloch neuron, and then leading on to the first neural network models. So the topics of experimental psychology are radically expanded since then. To pick up from the prize talks yesterday, I don't think the experimental psychology group would have ever thought about work on children learning mathematics or on recognition of emotions, but the overall perspectives of the mind as an information processor and of integrating understanding from cognitive, behavioral and neurophysiological levels, I think is probably stronger than ever. So to celebrate the EPS's 75 years, it would have been nice to bring in some of those original 8 members, but that's not very practical. So instead, we have brought together a list of people who have had a lot to do with the EPS over the years and will cover the breadth of topics that now characterize our discipline. And we wanted also to bring together people from different career stages, and so selected people who'd won different EPS prizes. So we begin with Aidan Horner, who will be talking about memory, who won the 5th prize in 2013. Lolly Tyler, language, Bartlett lecturer in 2015. Mike Burton, faces, the mid-career award in 2012. Nikki Clayton, cognition in the J, mid-career award 2013. Pat Rabbitt, aging, Bartlett lecturer in 1991, the previous president of the society. And Jennifer Cook, social cognition, 5th prize 2014. Before we begin, let me explain a little bit how the Q&A works. You'll see on your screen that you have a chat function, that's just for chat, which won't be not putting in a question that you want to be asked at the end. If you've got a question, you can type it into Q&A, or you can, as usual, raise your hand, and we will do our level best to notice that your hand is raised at the end, and we'll unmute you and you can ask a question. So our first speaker today is Aidan Horner, with a talk along with Lisa Henderson and Nora Andermane from the University of York. Title is Forgetting Across a Hierarchy of Episodic Representations. And Aidan, you can share your screen and go.
Thank you very much. Right, so let's get this up. And John, can you just confirm you can see it?
I can see it very well, thank you.
Great, thank you for that. And thanks everyone for coming. Feels slightly daunting to be speaking in this sort of symposium with all the other previous prize winners and stuff, and as soon as you said 2013, it suddenly made me realize how long ago the 5th prize was. But hopefully I'm going to tell you about some of the recent work we've been doing on forgetting, and persuade you that some of the work we're doing is just as interesting as the previous work to which the EPS decided was worthy of a prize. So this is work in collaboration with Lisa Henderson and Nora Andermane, and also Bardo Jørgensen, who I'll flag up in a second. And what I want to talk about is forgetting. So firstly, I want to acknowledge all the people in the lab over the last few years who have contributed to not necessarily this work, but other work that I won't be presenting today. But as I said, today I really want to flag up the work of Bardo Jørgensen, who's a PhD student in my lab who started this work, and Nora Andermane, who's done the more recent work that I'll get onto in the second half of the talk. So I couldn't really talk about forgetting without starting with Ebbinghaus. So the general point I want to make here is that a lot of people assume that forgetting is more or less a unitary process. So when it comes to forgetting, we think of the Ebbinghaus forgetting curve as a sort of exponentially decreasing curve. And we assume that that applies to all types of stimuli, all types of memory. So this is just the classic Ebbinghaus data that is plotted on a log curve, and that's why it looks linear in nature. And equally, there's been huge debates in the literature about whether forgetting is caused via interference or decay, so one or the other. So this is a classic study by Jenkins and Dellenbach in 1924. And it's some of the first work to show that there's a difference in retention dependent on whether you sleep or are awake between encoding and retrieval. And they showed that there was less forgetting during sleep, and they took this as evidence for an interference as opposed to a decay type account, because they presumed if it was decay, it should be relatively uniform across time. So you can see that there's at least an implicit assumption here that, again, forgetting is unitary in nature. So the results of our study as a whole indicate that forgetting is not so much a matter of decay of old impressions and associations, it is a matter of interference, inhibition, or obliteration of the old by the new. So they're referring to forgetting here. They're not saying forgetting for the syllables that they happen to be retaining or the experimental setup they've used. They're talking about forgetting as a, by and large, a unitary construct. However, more recently, I think there's been some interesting data to suggest that forgetting isn't a unitary process. So this is an interesting study by Fisher and Radvansky in 2019. So here they're plotting, again, the Ebbinghaus forgetting curve with Ebbinghaus's traditional data, and you can see that classic sort of exponential decay function. But what they pointed out is that in some of the experiments they ran and in some of the previous literature, there are situations where actually you get a linear decay function. So it's not that the rate of forgetting changes, it's the function itself changes. So a lot of the time we assume this in studies, but it might not be true, and there might be situations where we see linear forgetting. And one of those situations seems to be when we form more coherent complex event representations. Now, this is an opinion or review by Sade et al. Back in 2014. So these are just sort of theoretical graphs, but they do have some evidence to, empirical evidence to back this up now. But their suggestion was that actually memories or forgetting can be caused by either interference or decay, dependent on the type of memory that's being tapped into and the underlying neural region that's supporting that. So they suggested that familiarity, which is by and large supported by the perirhinal cortex, might be more susceptible to interference, so high versus low interference, and not by delay. Whereas recollection, which is a largely hippocampally mediated process, might be more susceptible to delay periods and not interference. And their argument was that, well, in the perirhinal cortex and neocortex in general, representations might be quite distributed and overlapping in nature. That means they're more likely to be susceptible to interference. Whereas in the hippocampus, we have more sort of pattern-separated sparse representations, so interference is less likely to play a role. Therefore, they're more likely to be forgotten via decay processes that might occur via sort of underlying neural processes like neurogenesis. Okay, so the key point there is forgetting probably isn't unitary in nature, and I'll come back to this point later. But what I want to do now is ask a slightly different question, and this gets on to Bardell's PhD work. So the question here is, how are complex events forgotten? So here we have some complex events that require you to learn a location, a person, and an object. So the example I always give is Barack Obama, so a famous person, in a kitchen with a hammer. And you have to imagine these three things interacting in some meaningful way. And then we're going to test your memory for those events later on. Now, presumably later on, you're going to forget some information. So accuracy between an immediate test and a delay period, after a delay period, is going to go down. But what we're really interested in is this measure called retrieval dependency. So Bardell and I distinguish between two types of forgetting. So in one situation, we might remember events in their entirety or forget them in their entirety. So we refer to this as holistic forgetting or non-fragmented forgetting. Now, this retrieval dependency measure is basically a measure of how likely you are to retrieve an individual element within an event, given you've remembered another element within that same event. So if you remember Barack Obama, you'll be more likely to remember Hammer for that event. Or if you don't remember Barack Obama, you'll be less likely to remember Hammer. Now, at immediate test, we should see a certain level of retrieval dependency, but the critical prediction here is if we see this sort of non-fragmented or holistic forgetting, we should see similar levels of dependency after a delay, despite the fact that there's been forgetting. Because your retrieval success here is still related to here, because you're remembering this event sort of in its entirety, whereas here, this whole event is dropping out, okay? So your retrieval, you forget Obama, therefore you're more likely to forget Hammer. Now, we contrasted that with what we call fragmented forgetting. So in this situation, the same amount of information is being forgotten. So in this case, there's sort of 3 elements, but it's all from the same event. Whereas in this case, there's 3 elements, but they're across 3 events. So accuracy goes down the same amount. But in this situation now, we break the relationship between retrieval success within an event. So that should mean dependency should go down if there's fragmented forgetting, because now your retrieval success for this element is less related to this element within an event. So Bardell tested this across a number of studies. So what I want you to focus on here is what I'll call the closed loop structure. So I'm not going to go into details of the differences here, but basically in this closed loop condition, across separate encoding trials, people are forming integrated representations of those 3 elements. So we're going to test immediately and after 12 hours. And you can see that some forgetting is occurring. And we see that across a whole number of studies as well. So here in experiments 2, 3, and 4, the delay was a week rather than 12 hours. You can see more forgetting is occurring. In experiment 3, we didn't test immediately. We just tested after a delay. So forgetting is occurring. The key question is what's happening to that retrieval dependency measure? Is it staying the same or is it going down? So here's the results from experiment 1. So here I'm plotting dependency, and 0 here means there is no evidence of dependency. In other words, if you retrieve Obama for a particular event, that tells me nothing about your retrieval success in relation to Hammer or Kitchen. Whereas anything above this 0 line means there is evidence of dependency, i.e. that your retrieval success for one element is related to your retrieval success of another element within that event. So what you can see is at time point 1 and time point 2, there is robust retrieval dependency, and there's no evidence of a decrease over time. So this suggests that the forgetting of these events might be more holistic and not fragmented in manner. And we see that across a number of studies. So experiments 2, 3, and 4 essentially show the same thing. Although there's a numerical decrease here, it's not actually significant. And if we do Bayesian statistics on this, we actually find more evidence in favor of the null. So evidence in favor that there isn't a decrease in dependency over time. So we took this as evidence for a more holistic form rather than a fragmented form of forgetting for events specifically. These results, though, are in contrast to some nice work by Tim Brady and colleagues in 2013 that asked a similar question, but with objects and their properties. So here participants are learning a series of objects, and then they're going to test your memory for those objects with these 4 alternative forced choice situations. And hopefully what you can see here is this particular orange juice is in a pint glass and it's full. So what they did was they manipulated 2 separate properties, the exemplar or the shape of the object and the state of the object. So whether it was full or half empty if you're a pessimist, I guess. So what they can do is perform essentially similar retrieval dependency analyses. So they can say, what's the likelihood of you retrieving the state given you've retrieved the exemplar or vice versa? And does that retrieval dependency decrease over time as a function of forgetting? In other words, is the forgetting of the state independent from the forgetting of the exemplar? And in contrast to our results, they show that there was a decrease in dependency over time. So they see a decrease in dependency over time, which seems to provide evidence for fragmentation for object properties. So we have a slight discrepancy between our results and their results, but obviously they're very, very different paradigms. So the key points so far are that forgetting isn't a unitary process. Forgetting can differ, the forgetting function can differ. It can be linear or nonlinear. And forgetting can occur via interference or decay, perhaps dependent on the type of memory or the neural region that's supporting that particular representation. And we can also track changes in retrieval dependency as a function of forgetting. And we seem to possibly have evidence for fragmentation of objects versus non-fragmented or holistic forgetting of events. So forgetting may occur at multiple levels in a representational hierarchy, and the nature of that forgetting may differ across those levels. So Nora, Andermane, Bardo Jørgensen, and myself a year ago decided to try to bring some of these findings together and outline a kind of tentative framework for thinking about forgetting at different representational levels. So here we're sort of outlining at least 3 separate levels. Obviously, they're not completely separate, but they're worth thinking about in terms of different levels and how forgetting might occur. So if we have a complex narrative that unfolds over time, that's likely to be represented within anterior hippocampal and ventromedial prefrontal regions. Now, within those narratives, there's likely to be more discrete episodic events, so that have, say, a single spatial context. And we think those are likely to be supported by the posterior hippocampus. And then obviously within an event, we might have individual items and their features. So those, in particular with objects, they're likely to be represented in the perirhinal cortex and more posterior ventral visual regions. Now, I'm not going to get into the details of all these 3 levels, but one of the key predictions we made was that there might be a difference between episodic events that are supported by the hippocampus and items and their features that are supported by the perirhinal cortex. And we predicted, in line with the sort of contrasting results of the Brady et al. Study and our previous study, that objects and their features are more likely to show fragmentation because perhaps the individual properties are represented independently. So the color and the shape of an object might be represented in independent regions of the neocortex, and interference in one of those regions and not the other might induce a sort of more fragmented form of forgetting. Whereas for events, they're more likely to be coherently represented in the hippocampus. They're more likely to be forgotten via decay processes that are more likely to perhaps be more uniform in nature and therefore lead to a more holistic form of forgetting. So that's what we set out to test. So this is all work now by Nora Andermane. And obviously what we wanted to do was bring the sort of object versus event level testing into a single experimental setup. So what she's doing here is presenting images of objects. So in the Bardo Jørgensen studies, they were all words on the screen. So now we're switching to images. But again, we have a particular location, a famous person, and an object. And we're going to test your memory for these event triplets in two separate ways. So the first way is trying to get at that event level representation. And what we're going to do is cue you with amongst 3 other foils. And these foils are objects that are related to other events, so other people in other locations. Oh, it's saying my internet is unstable. So can people still hear me okay?
Yes, Aidan, it's fine.
Yeah,
okay,
it's
great.
fine, not too bad.
So we're going to test your memory for these events across multiple retrieval trials. So we might cue with a person, get you to retrieve the object, but on another trial, we'll cue with the object to get you to retrieve the person, etc. And we'll measure retrieval dependency at this event level immediately and after a delay. In a separate set of participants, though, for experiment 1 at least, we tested object memory. So here we're still going to give you one of the cue, but now we're going to test object memory more similar to the way the Brady et al. Study did. So here they have to pick out the collapsed or the squished yellow duck and ignore the other 3 ducks. So here in these experiments, we have a color and a state for each individual object. So we can look at retrieval dependency for the color relative to the state across time. Okay, so T1 and T2 are within subject design, so we test immediately and after 3 days, but the object versus events is between subject here. And you can see that this is plotting retrieval accuracy. Accuracy goes down over time, so forgetting is occurring. The key question, obviously, is what happens to dependency? So here again, I'm plotting dependency for T1 and T2, so immediate versus 3 days for the events. And anything above 0 is significant evidence for retrieval dependency. So you can see that there's robust dependency at time point 1 and time point 2, and consistent with Bardor's original findings, we don't see any decrease in retrieval dependency at the event level. For the object level, we see significant dependency at time point 1 and time point 2 again, but now critically, we're showing a decrease in dependency over time. So this is consistent with the Brady et al. Study, suggesting that perhaps object properties fragment over time as a function of forgetting. So this was great. We've confirmed our hypotheses, but what we wanted to do was replicate this, and perhaps that was a mistake because then things started to get a bit more complicated. Okay, so what we wanted to do in experiment 2 was test this all within subject design. So we're now testing both objects and events all within subjects. And what we had to do was make some design choices to make sure that the events were off ceiling at time point 1 and the objects were off floor at time point 2. So T1 now is actually an hour after encoding, just to bring accuracy down a little bit. And T2 is after 2 days rather than 3 days. But you can still see that there's robust forgetting in both cases. In terms of events, well, we replicate that there's no decline in dependency over time, so robust dependency at time point 1 and time point 2. But now, somewhat confusingly, we see no evidence for a decrease in dependency for those object features. So this is in contrast to the Brady et al. Study and our previous experiment 1. So we have a discrepancy here specifically for the objects. The events now are very, very consistent. We've replicated this across 2 experiments in Nora's studies and, of course, the 4 experiments in Bardor's study. So we're fairly confident at the event level now, but something odd is going on at the object level. And one of the things that differed, obviously, as I've just explained, between experiment 1 and experiment 2 was time since initial encoding. And in particular, we tested in experiment 1 immediately after encoding and experiment 2 after an hour, just to bring down event level performance. So what we thought was, well, one of the reasons we might not see a fragmentation here is that actually that fragmentation process is very, very rapid, as in it occurs over the first few hours, and we might miss out on that decline in dependency by testing after an hour. So what we did in experiment 3 was we only tested objects, but now we have 3 time points. We have immediately after encoding, after 5 hours, and after 2 days. And the reason we picked 5 hours is because we ran another undergraduate study that used immediate and 5 hours for objects and did show decay, fragmentation over time, so a decline in dependency. So if we plot dependency now at time point 1, so immediately and after 5 hours, we do show a decrease in dependency. So this seems to suggest that maybe that fragmentation process is quite rapid for objects. So then we thought maybe it will stabilize over time. But somewhat confusingly at the moment, at least, so we haven't actually finished testing on this experiment, dependency seems to go back up again at time point 3, so after 2 days. So the T1 versus T3 seems to replicate the effects of experiment 2, but T1 versus T2 seems to replicate the effects of experiment 1. So this is a little bit confusing. We're still trying to get to the bottom of this. So one of the ways we tried to look at this was plotting the data in a slightly different way now. So not just looking at that overall retrieval dependency, but looking at retrieval accuracy for 4 different situations. So for any given object, they can either remember both the color and the state. They can remember the state but not the color but not the state, or neither the color or the state. And I'm plotting here those 4 options for time point 1, so immediate, and time point 2 after 5 hours. And what you can see is the sort of joint retrieval, so both of them being retrieved, go down, but you can only see an increase in these sort of partial remembrances, so state but not color and color but not state. So these go up, but the neither responses don't go up. So what that's suggesting is that participants at time point 2 seem to be forgetting the color or the state, but not both of them. And that's perhaps why that retrieval dependency measure is decreasing. However, after 2 days, if we compare T1 versus T3, well, now you can see it's much more even across these 3 sort of forgetting options, basically. So the neither responses seem to catch up with the partial remembrances at time point 3. And that might be why there's a rebound in that retrieval dependency measure. So why is this occurring? Well, I think one possibility, not necessarily the only one, is that maybe the forgetting of objects is non-fragmented, but it's temporally separated. And so in other words, if you forget the state of a particular object at time point 2, you're more likely to forget the color later on in time at time point 3. Okay, so to conclude, episodic events are forgotten in a holistic manner. So we see no evidence for decreases in dependency for events across multiple delays using words or images. Object properties seem to be forgotten in a less holistic manner. So things are clearly more complicated here and we haven't quite got to the bottom of it. So we do see decreases in dependency across delay under some conditions. In experiment 3, it suggests a possible solution in that we might have non-fragmented but perhaps temporally separated forgetting. But ultimately, I think changes in dependency at the object level are likely to depend on multiple factors. So perhaps the lag between study and test is critical. We've shown that with experiment 3 to some extent. The encoding time and task might be critical. And perhaps at a more theoretically interesting, it might depend on whether object features are hierarchically or non-hierarchically represented. And it might depend on situations where some of the object features actually become embedded or critical to the events themselves. And then those object features might actually become embedded within the event representations that are outside of the perirhinal cortex and more likely to be hippocampally mediated. But ultimately, I hope I've persuaded you that forgetting is interesting and that it's it's not a unitary process and is likely to vary both quantitatively and qualitatively across representational levels. So I'd like to thank the funders, past and present, for funding some of this work, and you for listening, and of course Bardor and Nora for all of the hard work. So thank you.
Thank you, Aidan. So now, as I said, you can either type questions into the Q&A or use the raise hand function, and we will unmute you.
Nobody's had their coffee yet, so.
While people think, let me ask a rather open-ended question, I suppose.
Sure.
Beginning really with the third aspect you haven't been talking about very much, that is this whole episode, extended episode memory. It's very striking that when, for example, suppose that you have forgotten the contents of a talk that you heard last week, and you can't get it at all, and just one little hint and suddenly the whole talk comes back. So very, very non-fragmented. And it then seems that every part of it, or any part of it in a way, is a very strong retrieval cue for everything else. And that makes it very interesting to me that you ever get a more fragmented picture, because it implies that one part of the object isn't really a strong retrieval cue for the other one anymore, that somehow they're not, well, I don't know, it seems surprising to me that memory would ever be that way.
Yeah, I mean, it's interesting because other people I talk to say, well, of course you forget some aspects of events and don't forget others, so it's likely to be fragmented. So I guess this is why we need to do experiments to some extent. I think the narrative level is interesting though, and is sort of underexplored. So I mean, people have done interesting studies looking at sort of videos or stories and showing that sort of gist-based memory tends to be forgotten less rapidly than the individual perceptual features. But as far as I'm aware, nobody's really looked at this measure of retrieval dependency, at least, to see the extent to which narratives are more holistically forgotten. So my sense is they're likely to be somewhat less holistic in manner than the sort of more discrete type events. So the example I tend to use is, a birthday party, just because I have experienced several children's birthday parties. It's a clear narrative that unfolds over time. But you're likely to move, say, between rooms or from, your living room to your garden. And it might be that you forget that you played pass the parcel in the living room, but you remember playing football out in the garden or whatever it is. So I think it's an underexplored area that needs to be looked at. But ultimately, I guess these experiments suggest that we can't just rely on our intuition, which is usually the case in psychology.
Thank you. Okay, so from the Q&A, we have a question from Daniel Yarn. I'll read it out and then you can answer. Beginning with a compliment, elegant studies and lovely talk. In your model of 3 different levels of representation, you suggested that objects could be forgotten in a fragmented way because features are represented in a distributed way across the cortex. Does that mean you predict that objects should be forgotten more holistically if they are defined by similar features?
I think the short answer to that is yes. So I mean, some of this sort of object work is built on some sort of on that Sade et al. Paper and opinion piece and work that suggests that it's likely to be interference that is driving forgetting for more sort of neocortically represented memories. But I think there are going to be, as I said before, I think objects are just likely to be more complicated. There are all sorts of context dependent situations and experimental situations where it might be more or less holistic. But I think the really interesting thing is how consistent the events are across time now. Like, we never show decreases in dependency for that. The objects are much more complicated and likely to vary, but events don't. And that to me is quite striking.
Okay, I think we have a question that came into the chat. Flagrantly disobedient member of the audience. Thank you for a fantastic talk. It was super clear and very intriguing. My question is about the role that relative distinctiveness similarity played in the results. The object laws were very similar to targets, perhaps more so than event aspects were similar to each other. Might this be important?
Yeah, yes. I mean, I struggle sometimes with the differences between the way we're testing memory for objects and the way we're testing memory for events. They're not as similar as I would like them to be to draw sort of strong conclusions, but the starting point was trying to at least have similar test situations to what we've done previously with events and what Brady et al. And others have done with objects, but sort of within a single stimulus set and experimental context. I think we could have done better, or we can do better in the future about trying to equate those test situations a little bit more carefully. And as you say, distinctiveness and similarity is something we probably need to pay careful attention to. But at the moment, the object results are kind of so complicated that we're still trying to wrap our heads around that before we can then do perhaps more careful manipulations of these different parameters.
Thank you. Okay, I think we have time for one more from the Q&A. This comes from Carolyn Patterson. I forgot to mention that the previous one, incidentally, was from Deborah Talmy. So from Carolyn Patterson, at some levels, it seems to make perfect sense that the hippocampal and the perirhinal systems should be sensitive to different factors in forgetting. But can you say a bit more about why you find this particular association that you and others do? P.S. Yes, elegant talk.
This particular, I'm not quite sure what you mean by this particular association. Are we allowed to unmute Carolyn if she's willing to unpack that?
That's probably a good idea if I could find her. Sorry
to put you on the spot, Carolyn.
Okay.
What I mean is that if interference for perirhinal cortex and decay for hippocampal.
So why it's likely to be decay versus interference?
Yeah.
So the Sade et al. Paper, and there's another paper, I can't remember, Hart is the first author, they came out similar times, basically make the argument that in the neocortex, the representations are likely to be more distributed and overlapping in nature, and that allows for more of an interference process because there's just more overlap and more interference when you're learning similar items. So that's the reason for the interference there. So then I guess you ask, well, why doesn't that occur in the hippocampus? Well, that's because the representations are thought to be much more sort of pattern separated in nature. So there's less essentially neural overlap between similar representations. So if you learn a new event that is remarkably similar in nature, it's less likely to be using the same neurons to represent it because of, say, pattern separation processes. So that sort of suggests that it wouldn't be interference. So then you ask, well, why might it be decay? Well, I guess one, because that's the alternative. But two, there is evidence to suggest that, say, neurogenesis in the hippocampus might sort of interfere with the retrieval of particular events, but in a much more sort of underlying neural circuitry way, as opposed to interference via encoding of new information. Those authors attributed or referred to that as decay. I think you could argue it is interference in a different way, in a much more sort of neural circuit way, as opposed to learning of new information. I hope that answers your question.
Okay, thank you very much, Aidan, and thanks again for your talk. I think it's time to go on to the next.
Thank you.
Which is from Lolly Tyler from Cambridge. Deep neural networks as explanatory models, vision and language. This is a recorded talk, which I believe we will start up right now.
Networks. And hello, I'm going to talk today about some of the work that we've been doing in my lab, focusing on deep neural networks and asking how well we think they function as explanatory models for cognitive processes. And I'll be doing this in the context of two domains, vision and language. To develop explanatory models of human dynamic cognitive processes such as visual object recognition or spoken language comprehension, we need to develop computationally explicit accounts of what neural computations support these processes and where and when they're located in the brain. Today, I'm going to consider how far DNNs for vision and language can be related to these explanatory goals. This means that we need to go beyond only matching input-output relations and try to relate the internal properties of DNN processes in any given domain to the underlying brain system supporting human performance. To do this, we need a well-developed biological model so that we can interpret the details of how the computational model works in any cognitive domain. We need this in order to bootstrap into other aspects of the cognitive domain of interest. Vision and language are very different in their relationship to DNNs. In the case of visual object recognition, we've got very well developed and detailed biological models of vision. Similarly, DNNs have proved to be useful for developing computational models of vision. But as I will argue, DNN accounts of visual object processing are limited in that they don't actually include higher level processes associated with the meaning of objects. Their focus is entirely on the visual properties of objects. But what is a model of object recognition worth if it doesn't capture the meaning of an object? Language processing is very different. Language is more challenging because there's no detailed and comprehensive biological model, and current computational models are motivated by engineering solutions with little reference to the human system. This makes it less straightforward to relate DNN language models to operations in the human brain. In my talk today, I'll discuss current research relating these two black boxes to each other, showing how DNNs may be profitably related to human neural solutions. Starting with visual object processing, we already have a well-established biological model of vision consisting of a hierarchy of visual processing in the inferior temporal cortex. This provides an important reference point. Along this visual pathway, increasingly anterior regions process increasingly complex visual information. Neurons coding for simple feature conjunctions are located in posterior sites, with more complex conjunctions of features in anterior sites. The perirhinal cortex at the top of the pathway contains neurons that can do the most complex feature integration, which enables the disambiguation of highly similar objects. So turning to computational modeling, there's a strong tradition in machine vision that reflects these theories of cortical organization. But there's more than visual processing alone going on along the ventral processing stream. Similar to the visual system, there's a semantic hierarchy along the stream, going from the processing of simple semantic features in posterior sites and more complex feature integration in more anterior sites, This underpins the semantic processing of visual objects and is essential for constructing the meaning of an object that we are viewing. This well-attested model of visual object processing enables us to make direct comparisons between DNN systems for object recognition and the real-time neural activity of human perceivers. This kind of approach shows that DNNs are very successful in modeling the early stages of visual processing in the brain, but fail to capture meaning-related processes. Let's look at some of the evidence for a conceptual hierarchy along the ventral stream. This slide shows the results of an Fmri study in which subjects named familiar visual objects like apple, car, rabbit, as they were being scanned. To test for different kinds of semantic information along the ventral processing stream, we constructed a variety of different kinds of semantic feature models, with some models capturing different kinds of category structure like animals or tools, while others captured more fine-grained properties of objects. We tested all of these models against the brain data along the ventral stream to see whether, as we expected, category information was associated with neural activity in the fusiform, and more object-specific, more fine-grained semantic information was associated with neural activity in the anterior temporal cortex. We tested each of our models against the Fmri data along the ventral stream using RSA, representational dissimilarity analysis developed by Nicola Krieger-Skold. As you can see in the slide, the results showed a gradient of semantic specificity along the ventral processing stream, with category information represented along posterior fusiform and object-specific, more fine-grained semantic information in the anterior medial temporal cortex, especially in the perirhinal cortex. We've seen that both vision and meaning are processed along the ventral stream, but one of the important questions that we still don't understand is, how does vision activate meaning? To address this question, we turn to computational modeling and ask, can DNNs explain how vision activates meaning? This slide shows a standard DNN processing visual inputs. The DNN consists of various layers processing different aspects of the visual input. For example, lower layers of the DNN better match early stages of visual processing, whereas higher layers of the DNN better match later stages of the stream. However, it's important to keep in mind that DNN models are designed to distinguish between objects and associate each object with its correct label. But there's no semantic content in the relationship between object and label. Unlike humans, DNNs don't activate conceptual information. To model the interaction between vision and meaning, we combined two computationally explicit models, a visual model and a semantic neural network. The penultimate layer of the DNN serves as input to the semantic model. And the semantic model consists of a recurrent attractor network where concepts are represented as patterns of activation across a network of semantic features. The semantic model was able to represent both shared category information and distinctive object-specific information. Through training, the model associated high-level visual information found in later stages of the DNN with concept features over the time course of object processing. And in the trained semantic network, semantic features activated gradually over the course of 20 time ticks. The activation of each feature was facilitated by feed-forward connectivity from later stages of the visual DNN, as well as lateral recurrent activity from other features in the semantic layer. The model therefore exploits statistical dependencies between high-level visual information and semantic features. Now, to test this model, we obtained Fmri data while people named common objects. We tested the output of the combined visual and semantic model at each layer in the model against neural data in key regions of interest. This slide shows the results of the Fmri object naming study. It shows brain regions known from previous studies to be involved in vision and semantics, And in each region, we can see the correlations between fMRI activity and the different layers in the DNN and semantic models. For example, early visual regions, for example, A on the slide, show the strongest model fit between fMRI activity and early visual stages of the DNN, and no significant fit of semantic models. In contrast, the perirhinal cortex, DE on the slide, shows significant correlations between fMRI activity and later layers in the semantic model. These results show that this approach is picking up differential visual and semantic effects associated with different layers in the DNN. It shows early effects of the visual model in posterior fusiform and later effects of the semantic model in anterior temporal regions. So this research demonstrates that DNN models of vision alone don't accurately capture the relationship between vision and meaning. That is, they do not provide a model of visual object processing. To achieve this, the visual DNN needed to be connected to a model of semantics. We found that in this model, vision activates meaning through the penultimate layer of the visual DNN, serving as an input to the semantic attractor model. In the attractor network, concepts are represented as patterns of activation across a network of semantic features. This kind of model enables us to represent both features that are shared with many other concepts, providing category information, and other features that are distinctive, providing object-specific information. And through training, the combined model associates high-level visual information found in later stages of the DNN with concept features over the time course of object processing. And it does this by exploiting the statistical dependencies between high-level visual information and semantic features. Vision is one of the best cases for DNNs, largely because there's a very well-developed neurobiological model. But to understand the relationship between the DNN and the biological model, it's important to know in as much detail as possible what the various computational stages of a DNN actually involve. And we can only do this by having a well-attested model of the human system. In our case, we had a specific question: how does vision support object meaning? And by relating a combined visual DNN and semantic attractor network, together with appropriate neurobiological models, we were able to at least generate some interesting hypotheses about how that might work. Now, in contrast, language is not like this. We are still dealing with two models that we don't understand, both the DNN and the human. We're now turning to some research that we've been doing using DNNs to help us understand the neurocomputations involved in spoken language comprehension. So first of all, for all of you who effortlessly understand language without thinking about it, a little bit of context to give you an idea of the complexities involved. This slide illustrates some of the properties of spoken language comprehension. So for example, that comprehending language involves accessing the meanings and the syntactic and semantic implications of each word as it's heard. It involves integration of each word into the developing context, processes of combination, and the rapid incremental activation and integration of multiple layers of different kinds of information, and the development of constraints of many different types that are placed on the upcoming speech. So we have multiple types of constraints, all converging on the interpretation of each incoming word. And in addition, the real-time computations involved in all these processes are very rapid, on the order of milliseconds. This requires that we use a neuroimaging method that provides measures of neural activation at millisecond temporal resolution. And MEG, magnetoencephalography, is the ideal method here, since it tells us both where and when, with millisecond accuracy, activity occurs in the brain. So why use a DNN, in this particular instance, LSTM, to understand the neurobiology of incremental language interpretation? Well, two important aspects of human language are that it is both incremental and predictive. The system is constantly predicting upcoming words based on the prior context. LSTM, as a successful current natural language processing system, is also incremental and predictive. So it may be useful in understanding the computations involved in language comprehension, because the LSTM output distribution can be used as a model of how the prior context constrains upcoming words in spoken language. And we can use this distributed model to test against neural language processing data. An important point here is that we really need to understand the computational history of prediction and the types of information carried in the prediction. A computational account that simply matches the output properties of the human system isn't sufficient. We need to understand the inner workings of the model and relate them to what we know about human cognition. Before I go on, I want to stress that what I'm going to talk about in the remaining time is work in progress and is illustrative of our approach rather than being a final story. In our experiments, we've used LSTM, standing for long short-term memory, which involves an input, in this case words, going through different layers, finally producing an output which is called the softmax layer. This model was trained on a 1 billion word corpus and has an input layer, 2 hidden layers, and a softmax output layer. This model is a powerful but poorly understood black box. As each new word is encountered, the model develops a set of probabilistic predictions of what the next word will be. The next few slides will give you an idea of these probabilistic predictions. The sentence so far is presented at the top of the slide. The manager explained that. And below you can see the continuations that the DNN produces in response to that input, together with their probability. You can see that both he and the are the words that are the most highly probable continuations, although there are many more that are possible with much lower probabilities. Here's another example of a probabilistic output distribution. You can see that the model is making sensible predictions at every word. And as you can see here, these predictions are not only accurate and make sense, but they're similar to those produced by humans. LSTM captures the sequential predictive structure of sentences. How does it do this? To discover what information it's using internally across the layers to generate predictions, we feed sentences to the LSTM word by word and correlate activity with different types of syntactic and semantic linguistic knowledge. We also relate LSTM activity with neural activity obtained while human subjects are listening to the sentences in a MEG study. The goal here is to bootstrap sufficient understanding of how the LSTM works to enable the LSTM to become an explanatory model which we can use in trying to understand the neurocomputational processes of predictions in human listeners. This research was conceived of and carried out by one of my graduate students, Hun Choi, a hugely intelligent young man. Our main goal here was to see whether LSTM prediction models can help us to understand the neurobiology of incremental interpretation in human listeners. So we generated models of LSTM activity at each layer of the model. We collected neural data in a MEG study of people listening to a variety of spoken sentences, and we tested the relationship between the models of LSTM activity and MEG data. This slide shows the types of sentences we used in the experiment. The first noun, the experienced walker, is likely to be interpreted as the subject noun of the sentence and combines with the upcoming verb chose. At this point, the sentence is ambiguous because the verb chose is associated with different probabilistic structures. For example, a direct object construction marked by the, or a prepositional phrase to, with a stronger preference for a direct object. And within each of these structural preferences, the semantics of the preceding words constrains the listener's preference for path rather than card. So what are the neural computations that are involved in these processes of ambiguity, prediction, and resolution? We turned to LSTM to develop models of incremental interpretation, which we could test against the MEG data. To understand the types of computations carried out in each layer, we first correlated activity in each of the layers with a variety of language models, syntax, for example, and semantics, to see what kinds of representation and predictions the DNN was constructing. We also correlated activity in each layer of the DNN with MEG neural activity. This slide shows the correlations between DNN activity in each layer and language models, a process which helps us to understand the kinds of computations that are being constructed by the DNN. The language models included various models of semantics, for example, the semantics of the subject-noun, semantics of the verb, and models which test for evidence of syntactic and semantic prediction. As you can see from the slide, at the beginning of the sentence, when the subject-noun phrase, the experience walker, is initially encountered, the LSTM0 initially represents the semantics of the subject-noun in the pale blue line, and when the verb is encountered, it represents the semantics of the verb, the dark blue line. There are no predictions. LSTM1, the second hidden layer, shows weaker subject-noun semantics and strong verb semantics as the verb is encountered, which persists during the following word. And it also shows evidence of prediction of the semantics of the complement noun at the article the. LSTM softmax, at this point, the LSTM has developed predictions about the kinds of syntactic structures that are possible or preferably associated with the verb, and it has also generated prediction of the semantics of the complement at the onset of the complement phrase the, and the semantics of the verb. So these results show similarities between the kinds of representations and predictions generated by the LSTM and those produced by humans as they interpret spoken language. The previous analysis helped us to interpret the kinds of computations generated by the DNN layers. We next look at the neural activity measured by MEG in each of the LSTM layers at each word throughout the sentence. We're looking for neural timing and location effects in the brain, and this timing is particularly important since we're interested in incrementality. Here we see how activity in each of the 3 DNN layers is correlated with MEG neural activity. We focus here on the subject noun, the experience walker, and its role in predicting the upcoming verb. Processing of the subject noun is accompanied by activity in all layers. In LSTM0, we see neural correlations in bilateral MTG and the angular gyrus brain regions, and also left frontal regions. LSTM1 shows only right hemisphere effects, again middle temporal gyrus and angular gyrus. And the softmax layer only shows activity in the right middle temporal gyrus. And since the MTG is frequently associated with semantic processing, we might speculate that different aspects of semantics are at this point being computed by the different layers. As we saw in the previous slide, softmax layer at the subject noun is involved in processing the semantics of the subject noun and predicting the upcoming verb. These processes correlate with neural activity in the right hemisphere middle temporal gyrus, brain regions known to be involved in semantic processing. In the lower panel of the slide, we see that comparable data from human subjects is associated with neural activity in the same right hemisphere middle temporal gyrus neural regions as the softmax layer. So far, we've been focusing on the correlations between activity in the LSTM and brain activity at the subject noun. Here we focus on the verb and the multiple probabilistic constraints associated with the verb in the sentence. When the subject, noun, and verb are presented to the DNN, we see in layer 0, LSTM0, the first hidden layer, there are weak and transient effects in posterior middle temporal cortex and angular gyrus. In layer 1, we see similar effects as shown in layer 0. So these 2 layers are only weakly interested in probabilistic predictions generated by the verb. In contrast, the softmax layer is strongly interested in the probabilistic predictions as shown by robust effects in bilateral frontal regions, left angular gyrus, and left middle temporal gyrus. These regions are plausibly involved in competition and or selection processes. These analyses, while only preliminary, illustrate that we can go beyond simply focusing on the output of the softmax layer when looking at the relationship between DNNs and language. We can test word by word throughout a sentence to try to understand the incremental processes in the real-time processing of spoken language. We have seen how activity throughout the DNN layers can be interpreted using different models of linguistic knowledge and relating activity in the DNN to time-sensitive neural activity and our current understanding of language and the brain. Making this level of detailed comparison between the computational operations within the DNN and the computational operations that we can now examine in real time in the human brain may help to build an evidence-based and hopefully explanatory understanding of dynamic cognitive functions.
Right. Thank you, Lolly, for that very interesting talk. I'm not sure that we actually have Lolly with us today. I think perhaps we don't, in which case we're going to have to skip.
No, I'm afraid not, John.
I was checking if she was in the attendees, but no. Okay, so we don't have Lolly Tyler with us. So let's give our mental at least applause. And we'll invite the next speaker, Mike Burton from the University of York, talking about recognizing faces, why familiarity matters.
Thank you, John. Can you see the screen?
Yes.
Yep. Okay, thank you. Well, thanks very much for the invitation too. It's really a pleasure to be able to take part in this celebration of the EPS 75th anniversary. So I'm going to talk about face recognition, and where I'd like to start is to think about what the problem of face recognition is. The traditional way to introduce this as a topic for psychological study is to point out that faces are all essentially the same. We all have the same basic layout of features. And so therefore, when we recognize people, we must be applying some very subtle analyses of key differences between faces. And this emphasis on telling faces apart actually represents the vast majority of the literature in the field. But over the last few years, my colleague Rob Jenkins and I have been studying a different component of this problem, which is how we tell faces together. If you recognize these two actors, you will have no difficulty in cleaving together the three different pictures of each of them and in cleaving apart the different people shown here. And yet, of course, the actual pictures showing each of the actresses are remarkably different. And this problem of within-person variability, how the same person can look different on different occasions, we've argued is a key part of the problem of recognition and an understudied one. In fact, it's not only understudied, it's sometimes deliberately controlled away, and we design experiments in order to get rid of what's regarded as noise in the different representations of the way a single person can look. So our argument has been that that's the wrong thing to do, and it's a key component of the problem. Now, these two components of the problem, telling people apart and telling them together, really clash in quite a difficult way when we face the problem of matching unfamiliar faces. If I show you these pictures and ask, is it the same person or not? If you don't know these people, it's probably rather difficult for you to say with any certainty, is it the same person or not? You don't know how to bring them together. Okay, so I gave the Mid-Career Award now 10 years ago, gave it in 2011, and this was my final slide, my conclusion slide from that lecture. And I thought it might be quite fun to revisit it and see what's changed. So the lecture was about this problem of within-person variance, how the same person can look different on different occasions. And I argued that if we could only study this in more detail and understand it, then stuff would be revealed that hasn't been so far in this field. Particularly if we look at matching faces, it's easy to match two faces of someone you know and difficult to match two faces or two pictures of someone you don't know. And we argued that that could be understood better by a knowledge of within-person variants. I also argued that it might help us explain why computer face recognition doesn't work and why we're ignorant of our own face recognition ability. So I'm just going to review what's happened in the last 10 years or so in each of these claims that I made 10 years ago, but it suits my narrative purpose to take them in a different order. So let's begin with the computer one. Why doesn't computer-based face recognition work? Well, 10 years ago, it didn't work. Today, it really does work. Of course, there are operational situations in which it doesn't work well, but lab-based computer face recognition now is remarkably accurate. And the reason for that is because of the increased emphasis on deep convolutional neural networks, often not motivated in a neurobiological way, in the way that Lolly was just describing, but nevertheless, they work fabulously. I'll give you a couple of examples. We've played with various DNNs and tried to see where they break, where we could find difficult face problems that they wouldn't be able to solve. So far, they solve pretty much anything we can throw at them. Here's a nice example where I took some stimuli from a lovely paper by Noyes and Jenkins on disguise, where people were sometimes trying to disguise themselves so they wouldn't be recognized by viewers, and sometimes trying to disguise themselves as another person, a specific other person. And these, for example, this picture at the top and in the top left are the same person, something that human viewers often find it very difficult to judge, but the DCNN that we were playing with had no difficulty. Possibly my favorite and least favorite example is this one. Because we were trying to look for difficult cases, we ended up looking at our own personal photographs. And these are two photos of me taken 40 years apart. Now, I think you have to know me pretty well to know that these are both me. And despite these sort of obvious differences, the inevitable aging differences and so forth, just the capture devices are different. Everything seems different about these two photos, and yet the DCNN can do it. And this is what we found typically, that DCNNs can outperform human viewers even looking at unfamiliar faces. And in some ways that makes them less interesting. That's another talk. Okay, so we got that one wrong. What about this one? Why is unfamiliar face matching hard? Well, I think we have made more progress here. And I think we have a reasonable answer, which is it's because faces vary in idiosyncratic fashion. Yes, every time you encounter me, my face will look different, and that applies to you too. But it turns out that the ways in which our faces vary are idiosyncratic to us. Now, that is important because if you have to learn for all the people you know how their faces vary, it explains to some extent why that is a very hard task when you have two pictures of someone unfamiliar and have to decide, is it the same person or not? Because for an unfamiliar person, you don't have the experience with that person to know how that person's face varies. Now, you can demonstrate this in a number of ways. I'll just show you a couple of them. One is that we can do statistical analyses of actual photos of the same person. So we often use Hollywood actors here just because you can get lots and lots of pictures of them. So what we do here is we take hundreds of pictures of an actor, Hugh Jackman in this case, and we perform some statistical analyses, in this case, principal components analysis, from which we can extract the dimensions along which Hugh Jackman varies. We can then do the same for some other actor, let's say Matt Damon. What are the dimensions along which pictures of him vary? And now we can ask, we have two spaces, are they structured in the same way? Are the dimensions on which one actor varies the same as the dimensions on which another actor varies? And the answer is, in the very early components, 1, 2, 3, you do have commonality. So early statistical analyses always pick up some key things like where the light is coming from and the angle of the face. But very quickly, once you get into about dimension 4, representations diverge and people have their own idiosyncratic ways of varying. Now you can show this in different ways. Here's one demonstration of this. Here we have a picture of the actor Hugh Jackman, and here we have a representation of him in dimensions or building blocks built from hundreds of pictures of Hugh Jackman, not including this one. And what you find is that you can actually get a pretty good representation using those building blocks of that photo. For those of you familiar with this technique, this is a representation in the first 60 principal components. What we can then do is take the same photo and say, well, can we build it in building blocks or dimensions derived from somebody else's photo? So here is a representation of the same photo built from dimensions derived from hundreds of pictures of Matt Damon. And there's a couple of things to notice here. Firstly, it's not as good a representation. It's not as good a representation as the original photo as this one. Secondly, it looks a bit like Matt Damon. And this is what you find. If everybody varied in the same way, it shouldn't matter where I get my statistical variation from, as long as I have a big enough sample to truly sample it. But it does matter. You are tied to the ways in which your face particularly varies in order to generalize new images of that person. Okay, so that's why we think this is difficult. We've recently been doing this with some even more extremely varying images. This is work led by Mila Mileva, and we were looking at some photos of people that we had photos of over 60 years. 1960s pop stars are a good candidate for this. Some of them who survive into the 2000s, you can get lots and lots of images of them. And what we find is if you do indeed learn the idiosyncratic variation of Brian Wilson, even just in the 1960s photos you can find of him, that does give you power to recognize him in much, much later photos taken 40 or 50 years later. But the key issue is being able to cohere together the variants of that particular person. Okay, what about the third topic that I picked up in the original lecture? Well, why are we ignorant of our own face recognition ability? This is more of a social question, really, I suppose, than a cognitive one. But why do we rely on photo ID? And we really do rely on it, sort of obviously at passports, but you know, students, if they want to buy a bottle of wine in a supermarket or gain entrance to a nightclub, they often have to show photo ID to prove their age. The UK government is currently introducing legislation to make it mandatory to prove your identity to vote using photo ID. Why? The one thing that all psychologists working in this field will tell you is that matching unfamiliar faces is difficult. Whoever has the job of matching you to your ID photo probably is not going to be highly accurate at that task. It's a difficult task. So why do we still use it? Why do we rely on it? And I think part of the answer there is because we overgeneralize our ability with familiar faces. So Andy Young and I have recently been arguing this. We have evidence all the time of how good face recognition ability is, because we just see people the whole time and recognize them, our colleagues and friends. But what we don't notice is that that same level of expertise, of excellent performance, does not translate when we're asked to perform a task with unfamiliar faces. Now, I said any psychologist would agree with that, and that is probably true now. But when we first started looking at unfamiliar face matching, there was actually some resistance. And if I go back to work in the end of the 1990s led by Vicky Bruce, she really demonstrated early the difficulty of matching unfamiliar faces, and people sort of didn't quite believe it. That's true both in the academic literature and in the applied settings where we were working with police officers and so forth. And people used to raise these objections and say, look, you're making unfamiliar face matching look harder than it is. And here are three of the ways they used to mention. Well, one is that we use participants who are unmotivated or inexpert. We use we use undergraduate students in the experiments who are doing it for a small fee. Police officers often actually object to this and say that is unlike the situation we're in. People point out that our lab-based studies usually use photographs. So you're asked to match two photos and say whether they're the same person or not. Whereas in many critical real life situations, you have a real person in front of you and you have to match that person to a photo, and surely that's easier. People suggested that we choose foils deliberately to make life look hard. So when we have two photos of different people, then maybe we've made a huge effort to find foils that look very, very similar to each other. And of course, one tries not to do that, but it's possible that that creeps in. Well, it turns out that none of these objections survive test. So let me just show you one example of that. This is work led by David White and Richard Kemp at University of New South Wales. And we were looking at Australian passport officers. We had access to a set of these officers, and we asked each one to sit behind a desk, and then they were approached by student volunteers carrying photo ID. And the officer just had to say, yes, you're carrying the true ID, that's you, or no, it isn't. Now, when they're carrying true ID, they were carrying a photo we'd taken 2 weeks earlier, but a photo that met passport criteria. So they were the types of photos that these officers were used to dealing with every day. Okay, passports, by the way, can be 10 years old, so this should be relatively easy. When they're carrying the wrong photo, we just chose a photo to swap with someone in the group of volunteers. Now, we only had 40 volunteers. We had 20 men, 20 women. We only swapped people within the same sex. And so you've really only got a little pool of 19 people to swap with. You know, we haven't got a chance to go out and find somebody's lookalike, is what I'm saying. Right, how did the passport officers do? Well, surprisingly not great. Here is the data. Each dot represents a particular individual officer, and the performance measure here is the overall matching accuracy. And what you find, firstly, is there is great discrepancy. There are large individual differences here, very large. And it turns out in other research, these are quite stable. Some people are doing perfectly on this task. Some people are doing really pretty terribly. A chance level is 50%. Overall, sort of average performance, however you measure that, it's around about 90%. And if you're tempted to think that's not bad, then just consider how this has to scale up. You know, in normal times, Heathrow processes about 200,000 passengers a day. Right? You don't want 10% errors in that sort of setting. All right, interestingly, people's performance does not scale with their experience in the job. So these people on the right here have been doing this job for 20 years, and some of them are really pretty poor at it. And some of them are excellent at it. And this is just what we find. These people on the right have been trained and trained and trained and trained, but the training doesn't seem to. So even people we expect to be able to do this can't do it as well as we might expect. What about this issue that I mentioned, that we overgeneralize our own abilities? Well, you can demonstrate that in a number of ways. Here is some work led by Kay Ritchie, where we showed people pairs of faces and simply asked them to say same person or different person. But we embedded in there some celebrities that we thought older people would recognize, and some celebrities that we thought younger people would recognize. And then when we analyzed the responses, we found exactly what we'd expect, that younger people were better at matching the younger faces and older people the opposite. But interestingly, our cover story here was, look, we're trying to develop a test for use with the general public. Now, for each of these pairs, please say how well you think the general public will do with these. And what the finding is that exactly echoes performance. The younger people thought that the general public would find the younger faces easier, and the opposite for the older participants. So the faces I find easy to recognize, I kind of assume you will too. You can demonstrate this in a number of ways. Here's another experiment from the same paper, where we tested people in 3 different universities, Aberdeen, Glasgow, and Kent, showed them pairs of faces, and we showed them those faces comprised actually staff from 2 of the universities, Aberdeen or Glasgow. And what you find when asked to do that matching is exactly the familiarity effect you always get. Aberdeen participants are much better at matching pairs of Aberdeen faces, who they know, than Glasgow participants. And the people in Kent who knew neither of the sets of faces are sort of bobbling along at about 70%, which is par for the course for these sorts of unfamiliar matching tests. What's interesting, again, is that we gave a cover story here. We're trying to develop a general test for use with the general population. How well do you think people in general will do? And again, people followed the faces that they themselves could do. So Aberdeen viewers simply think Aberdeen faces are easier to match than Glasgow faces are. And the opposite is true for the Glasgow faces. So there's this overgeneralization of one's own ability. And this crops up in other areas of psychology, of course it does, but it's neatly demonstrated here. Okay, so come to the final one then. Why is familiar face matching or recognition easy? And I think that this really, in some senses, represents quite a failure of our field, because this is where many of us start. How do I recognize my friends? And I think we're still quite a long way from understanding that. And the vast sort of field of face recognition researchers often don't address this question, of course. We do know some things about this. We know that learning face variability helps. If I'm to learn a face, it's a good idea to learn it over a big range. So to find pictures of the person I'm trying to learn with different hairstyles, in different lighting, over different times and so forth. That helps. We know that semantic and contextual support helps. Of course it does. Of course it does. Of course it's easier to learn a face if you get some information about the person whose face it is, than if you just simply have to remember the visual aspects of the face. Of course you do. We know that the effect of familiarity seems to be quite general across different ways of becoming familiar. So in some recent work led by Holger Wieser in Durham, we've been looking at ERP signatures of different types of familiar face. And you can, in the area that ERP discriminates between familiar and unfamiliar faces, which is about 250 to about 600 milliseconds, you can pick up some qualitative similarities between responses to faces who the viewer knows personally, who's a celebrity that everybody knows, who's a celebrity that the viewer loves, who's a celebrity that the viewer hates. All of those different types of celebrity, types of familiarity give rise to qualitatively similar ERP responses in contrast to unfamiliar faces. But there's lots we don't know. And I won't go into the details of this for time, but I think we are going to need, I think now, to understand familiarity by stepping out somewhat of what has become rather a silo of face recognition research and look at other domains in cognitive psychology where similar things might be happening. Often in face recognition, we contrast classes of objects with representations of faces. So what's the similarity between recognizing faces and being able to recognize certain objects, or certain objects that you're skilled with, or whatever it is? But I think the issue of familiarity of specific individuals is not really fully understood, and we have got to learn a lot from other parts of the discipline. So for example, if we step away from the visual sides of it, what is it that's in common between my recognition of a friend and my recognition of a tune that I happen to know, or my recognition of a poem that I happen to know, by contrast to all those that I don't? And my prediction is that this will become a key part of the field over the next decade. Okay, so this is now the second time that I've finished an EPS talk with the same slide. These are the 4 things that I promised we'd make progress on if we knew more about within-person variants. And I think we've made progress in a couple of them, not so much in the other 2. I've sort of represented that with ticks and crosses, the size of which represent my confidence in the claim, I think. But as you see, there's plenty that we got wrong and plenty that there is still to do. So I think it is still worth saying, because it's still not universally taken across the field, that variability between faces of the same person isn't a nuisance to be controlled away, but it is one of the key aspects of understanding familiarity. There are many more. Okay, thank you very much.
Thank you. So, same as before, we'll begin with either raising your hand or putting questions into the Q&A. And here comes first question from Andy Willis. Andy Wills, Thanks, Mike. Interested that DCNNs are so good at face recognition when they're really brittle on object recognition more generally, e.g. single pixel shortcut learning, texture rather than shape bias, poor out of training set generalization. The particular company I think you used, FaceSoft, seems to have folded. Is there any reference I can look at on this?
As I understand, it didn't fold. It went behind a wall that you are no longer supposed to ask it about. So I may be wrong about that. It's very hard for me to stand here and say, you know, DCNNs do this in general or do that in general. And of course, it's impossible to say that. What I can say is that they solve some very difficult parts of face recognition that humans really find it difficult to solve. So it may be that in trying to, as it were, trying to trip them up, trying to find where the difficulties are, one focuses on things that are sort of egocentrically difficult for humans, and that's where the problem lies. Now, I know that Peter Hancock in Stirling has been doing some comparison of human errors and DCNN errors for certain networks, and is finding some, not complete, but some correspondence between those two, and that may be where there's room to do this work. So please don't take my kind of being wowed by some of these examples as a general statement about DCNNs, because of course I haven't done a full analysis of all the available ones.
Perhaps while we wait, I'll ask a question. So your account of idiosyncratic sources of variability makes me immediately think that you might develop the following idea, that for each person you've got to learn which features are constant and therefore you can rely on them to identify that person, and you've got to learn which ones are vary, so you should throw them away. You've obviously emphasized the second of those more than the first one, but does that account really match your, I can't quite get my head around whether that matches your principal components, where you might just think what you had to learn was which PCs to look at and which PCs not to look at for Matt Damon, but it sounds as if there's more than that.
Yes, I mean, I completely agree with you that the way that you characterized it is exactly as we'd say. It sort of teaches you for each individual person which ones you can throw away and which ones you have to focus on. But I think that is something that the sort of key about that is that it means that you are stuck in learning a new person. You've got no other experience than just, you know, having to do the groundwork. It's like sort of vocabulary learning. You have to learn it. And I think that the contrast I'm trying to make here is with a view that we are somehow generally experts at face recognition, because this is an idea that was popular for quite a long time and much argued about, that we somehow we bring to the world our ways in which we understand faces work, and so that we can apply that when facing new people. And I think that just doesn't work. But no, I entirely agree with the way that you put it at the beginning.
Okay, thank you. I think I am not seeing any more open questions. Oh yes, I am. From a name I can't fully interpret. How do DCNNs predict other race recognition effects?
Well, there's a very, very long-standing statistical proposal for explaining other race effects, which I think holds true now as it did when Alice O'Toole introduced it 20 odd years ago, which is that the statistical regularities of your sample are the ones that you apply to your new input. And so if you have a sample that samples over the range of variance that's relevant to a restricted set, that won't generalize well to another set, and that is Alice O'Toole's explanation of the other race effects. And I think it's a pretty solid one now, and will be true of any statistical analyses.
All right, thank you. Well, we've reached 11 o'clock, which is our target time for the first half of this symposium. Again, thank you very much, Mike, and to Lolly and Aidan for their talks, the first part of this symposium. The next speaker in this symposium, Nikki Clayton, who's talking about Jays and magic.
Well, thank you very much indeed. Psychologists have become increasingly interested in using magic effects to explore blind spots in seeing and roadblocks in thinking. And recently, my colleagues and I have suggested that similar techniques could possibly apply to non-human animal observers, especially those such as jays, and there's a Eurasian jay pictured on this title slide, that watch where other birds have hidden their food caches and use this information at a later date to attempt to pilfer these caches. What I'd like to do in my 20-minute talk is to briefly review the caching and pilfering tactics of the jays, and then explore how we might be able to further our ways of thinking about these issues using a magical framework. Now, although there's a distinct advantage to caching because food can be hidden for future consumption, particularly if there's an abundance of a food supply and so far more food than you could eat right now, this behaviour is necessarily competitive and risky because other individuals can also pilfer the caches. And indeed, up to 30% of caches are lost each day to pilfering competitors. And here you can see a couple of examples. The cartoon on the left is of a magpie, and the bird on the right is a California scrub jay. Now, we call this pilfering behaviour rather than cache theft or stealing, simply to make it clear that we're avoiding any cognitive attribution of an understanding of own versus other property. But we should also add that this behaviour isn't fortuitous. We know that jays, such as the Californian scrub jay pictured here and the Eurasian jay shown in the title slide, will watch or observe where other birds have hidden their stashes of food and then rely on their observational spatial memory to recover these caches at a later date when the cacher has left the scene. Now, perhaps the most notable point to make here is that jays play the role of both cacher and the role of pilferer. So an individual bird does both, it caches and it pilfers. And consequently, this has led to the evolution of a suite of cache protection tactics, which the birds use when caching to minimise the likelihood that their caches are stolen by others. And here are some examples. So we've shown experimentally that they will use shade to degrade information. By that I mean that if you give them the choice of two places to cache, one in the sun and one in the shade, the birds will preferentially cache in the shade only if other competitors are watching. If they cache alone or when their mate with whom they share the food is watching, they're happy to cache in both the sun and the shade. They also remove information by caching behind barriers. Again, we've compared observed and in private conditions. They use distance, preferring to cache as far away from the competitor as possible, whereas if they're caching in private, they're happy to cache in near sites as well as far away ones. They're also able to keep track of which particular individual bird was watching and listening when, so they show a preference, for example, to cache in quiet places if others are listening but cannot see because their view is obscured by a barrier. If others can see, they are more likely to cache in the noisy sites as well as the quiet sites. And the cartoons here, which were drawn by one of my PhD students, Scott Stevens, show scrub jays engaged in this behaviour, but we have replicated all these effects also with the Eurasian jays. But they don't only use the cache protection tactics at the time of caching, because typically they cache food and then recover the food at a later date. It could be a few hours later, it could be many months later. But sometimes, rather than recover their caches to eat them, they recover them to move about. And perhaps in one of the most compelling cases of social cognition in these birds, we showed that they move caches at recovery when potential pilferers have left the scene, and that this behaviour isn't hardwired. Turns out that it's only experienced thieves. It's only birds who have had the experience of pilfering other birds' caches that do this. Naive birds don't do it. And the experienced pilferers are moving the caches that other birds had seen them cache at the time of caching. The recovery behaviour is always done in private, so it can't be attributed to the presence of another bird looking. Now, there are a number of interesting parallels here when it comes to the way in which they do these cache protection tactics that are reminiscent of some of the tactics that magicians use. So if they come to recache in private when no other birds are watching and no other birds had previously seen them cache, you can see the waxworms, these are the Belgian truffles of the jay world, in the bird's beak. And it's easy for a human observer watching over a webcam to see exactly how many caches are being moved and where they're being relocated to. But that's not the case if they're recaching in the presence of an observer bird. You can see here that it's very difficult to know exactly how many caches were moved. And indeed, some of them are just fake moves, hence the idea that it's sleight of beak, akin to some of the magic techniques that magicians use when engaging in various forms of sleight of hand. And they also have secret pockets, as you might see here. This is the little throat pouch. I'll indicate it here by the arrow, and you can't really tell whether there's anything hidden in there or not. In this particular case, there were 7 waxworms hidden in that pouch, but you can't really see whether there's anything hidden. And even if you detect a slight swelling, you can't possibly tell what was hidden. So there seem to be some interesting parallels between some of the tactics that the birds use to conceal their caches, such as secret pockets and sleight of beak, and the technique that magicians use in their magic effects. And to explore how we can further our ways of thinking using a magical framework, in the rest of the talk, I'd like to focus on some work we've done recently that was published in Proceedings of the Natural Academy of Science, and which was a follow-up on a paper we published last year in Science. And that's exploring the perceptual inabilities of Eurasian jays using magic effects. And we performed 3 different types of magic effect. They're called palming, French drop, and fast pass. And I'll explain exactly what they are in a minute. The point is that they rely on different cues and expectations, but all of which mislead human spectators into thinking that one object has or has not been transferred from one hand to another. And here you can see Eli Garcia Pellegrin, my PhD student and first author on the PNAS and science paper, performing one of these effects. And I should add that it's quite interesting that Eli and Clive Wilkins are both magicians. Clive's a professional magician, a member of the Magic Circle. Alex Schnell, my postdoc, and I are not magicians, but we know a little bit about corvids. So each of the jays was then trained to peck the experimenter's left or right thumb to indicate which hand they thought the food was in, having watched the experimenter perform the magic effect. And here you can see J-Lo, who just watched Eli performing the French drop effect, and here you can see her pecking Eli's left thumb. Now, note that these 3 different techniques rely on different cues and expectations, all of which mislead human spectators into thinking that the object has or hasn't been displaced. And here are the 3 different kinds. So in a palm transfer, the worm or the coin, if it's performed on a human person, is swiftly secured between the fingers and therefore appears to be transferred from the left hand to the right hand or vice versa, but actually it's retained in the initial hand. In the French drop, it's similar really, but the finger and thumb are used to fake a transfer, but actually the worm is dropped into the initial hand. The fast pass is slightly different. Of course, it still involves hands, but in this case, the worm is swiftly passed from one hand to the other, and it's actually concealed in the alternative hand. And magicians use these concealment techniques like palming and French drop to mislead the audience into thinking that the object of attention has been transferred from one hand to another, when in reality it has not. And for these tactics to successfully fool an observer, of course, the spectator has to have an expectation of the outcome of making certain hand movements. In other words, they have to anticipate where they think the item, the worm or the coin, will end up. So to be fooled by palming and French drop techniques, the spectator inherently needs some knowledge regarding the motions they're about to observe and the typical outcome. Because without this primordial expectation, there'd be no preconceptions for the effect to rely on. The fast pass requires a slightly different understanding in that the modus of operandi relies on the fast motion of the effect, not on the human's inability. So on the human's inability to spot the movement in which the object is quickly transferred from one hand to another, rather than any understanding of how the hands work. So we wondered whether the Jays would be fooled by the fast pass because it requires this expectation of fast movements, but not by the palm transfer and French drop, given that they don't have hands themselves or anything that resembles a hand. And we tested 6 Jays. And this is what it looked like when we did it for the Jays. So here you can see the palming technique. And there you can see that Stuka got that one correct. So she wasn't fooled by the effect. Here you can see the French drop technique. And there, if you look at the thumb, pecks the thumb, and again, she gets it correct. Here you see the fast pass technique. Now she's got it incorrect. She didn't get the worm. And here you see another one in the other hand. So now it's right to left. Again, she gets it wrong. So we tested 6 Jays, 3 males and 3 females. And what we found is that they're not deceived by palming and French drop effects. In other words, they pick the correct thumb and get the food. And that's something that requires them to expect an object to move between the magician's hands. However, they were misled by the fast pass effect. Here you can see the percentage of correct responses. So they were great for palm transfer and French drop. In other words, they weren't fooled by magic effects, but they were rubbish for fast pass. We did the same thing for humans, because it's one thing for magicians to say that these are reliable effects that they use for their audience, but we wanted to make sure that in testing our human subjects, they had exactly the same view as the Jays. So this is what it looked like. Here Eli's doing a French drop, and in fact, that's the view you would see as a human being, and it's all done on the web online, and you even saw a Jay come down and get it. And you see that for the humans, our human subjects were fooled by all 3 effects. So back to the beginning, this is the first study to investigate how another taxon perceives magic effects and relate it to their natural biology and ecology of caching and pilfering behaviour. And magic effects could provide an insightful methodology to investigate blind spots in seeing and roadblocks in thinking, both in human and non-human animals. And we hope offer unique opportunities to highlight the cognitive constraints in diverse animal minds and similarities between them. For example, asking to what extent the reason that the French drop and palming didn't affect the birds was because they don't lack hands. So would we find that some other animals that do have hands, some primates, for example, are also fooled by that? So with that, I'd like to thank my collaborators in the lab, and of course my feathered friends, and all of you for listening. Thank you.
Thank you very much, Nikki. And as before, questions, you can either raise your hand or type them into the Q&A. So we have a question from Federica, and I believe she's been unmuted. Off you go.
Thank you very much, and perfect pronunciation of the name. Thanks. Well done. I have just a question for clarification. The first two types of trick, so the palm transfer and the French drop, is it correct that the object remains in the initial hand with both of these?
That's correct. And I should add, because you're probably going to say, but surely in the third effect that you did, the object is transferred, we had a number of controls. Because I only had 20 minutes to present the data, I haven't explained the controls. But we do have one control conditions where it either remains in the same hand, in the case where normally it's transferred, and control conditions where it remains in the other hand, in the conditions where it isn't, and slow versions where it's easy to detect, so that we were able to show that it isn't just because of whether it's in the same hand or moves to a different hand.
That was exactly what I was going for. So the bird is not always just pecking the hand where the worm originally was seen.
That's correct. So we have control conditions, and we have what's called slow pass conditions, where we do it in slow, like slow-mo on a video, where you can literally track all the changes. And in those cases, both the humans, spectators, and the birds don't make the mistakes. They're not
fascinating. Fooled. Thank you very much.
No problem.
Okay. So now we have a question from Alex Kachalnik. You're still muted. Okay. No, still muted.
Now probably it's working, okay?
Yeah.
Nikki, good, yeah, great talk, and good to see you. And
you too.
One question, you frame the problem of the J in terms of expectations of what hands could do and what people could do, which is great. But the J could also form expectations on the basis of physical regularities, which are true for Js and for people, even if they don't have hands. For example, if you let a worm drop from one hand to another, and you could see the movement of the food from, say, a higher hand to a lower one, but then the correct answer is in the previous one, violating what you have seen physically. Would they actually get it right or wrong? I mean, do they understand the physics of the problem, or is correct to think only in terms of expectations?
We don't know, Alex, but thank you for reading my mind, because that's the next experiment we want to do. because we don't know yet, but it is, you know, it is easily testable. The only other thing we've done is one where there's food under a cup, and they either expect a boring peanut or a tasty piece of cheese or worm, and we had to use 2 preferred foods because different birds in that study have different preferences. And as in the Franz de Waal kind of experiments, if the food that they expect is there, everything's fine, and they just take the food. If the food they don't expect is a pleasurable surprise, they're expecting a peanut, they get some cheese or a worm, everything's fine. If they get the boring peanut when they were expecting a worm or cheese, their crest raises up and they chuck the cup at the experimenter. But we haven't done anything yet with looking at the causal physicality versus expectation, but that's what we want to do next.
Thank you.
Okay, well, if there are no more questions for Nikki, I'd like to thank you again very much for contributing to this symposium, and we will reconvene at 11:45 for Pat Rabbitt.
Thank you very much, everyone. Thanks for listening. Thanks for hosting, John.
Well, welcome back, everybody. I think we can proceed with the last two talks of this symposium. They're both recorded. The first is from Pat Rabbitt, the University of Oxford, 50 Years of the Experimental Psychology of Human Cognitive Aging.
I'm extremely honored and surprised to be giving this talk at this anniversary of the EPS. Had I known I'd be around when the EPS was so old, I would have thought differently about it. As it was, I suppose I must be one of the few people who remember it when meetings, particularly in Cambridge, used to be of about 12 people. It seemed very much more because of the monstrous egos in the room, principally Tony Deutsch and Stuart Sutherland. And the EPS has been praised and blamed for many things, blamed particularly by Hans Eysenck for interrupting his meteoric progress to the head of his profession in Britain. But I've dealt with this in a paper in Cause, Introduction to Eysenck's Life. The only thing I have to say is that both Dorothy and I, like so many hundreds of others, have found that the EPS was a constant source of stimulation when we were isolated in our departments and had few people in our subjects to speak of. It was a reliable source of interaction. And to use the phrase often used about it in those days, that it was a society for self-advancement and the consumption of alcohol, is, I think, unfair. Okay, I'll get on with this. I'm dating this exploration of the study of cognitive aging in the last century and this from the 1980s. Jim Burton had two ideas, basically. One was that neurophysiological indicators, at least sensory and motor indicators, would predict your age status. And he noticed that age predicts loss of sensory and motor efficiency. So if you're measuring these things, then if you take age changes in hearing, vision, and balance, partly they will be caused by peripheral changes in the eyes, ears, etc., cornea scratching and loss of otoliths and earwax even. And they may also in part be caused by brain changes, but we can't know this unless we also have and include in our studies some direct measures of the extent of what brain changes you achieved at the time when the measurements were made. But his second idea was extremely popular and extremely widely taken on board. He was looking for whatever functional behavioral characteristic he could measure was most sensitive to age-related change, presumably in the brain. So the second thing that Burton did in his lab with his associates Jack Botwinick and Joe Robbins was to test people on various single occasion lab experiments. And of all the measures he took, he concluded that decision speed is the most sensitive index of age-associated cognitive change. This is generally accepted, but the question was, how do we take this further? How is it more than a marker for your cognitive status? Well, a third guiding idea was from Nathan Schock, who in the 1950s started a long-lasting Baltimore longitudinal study. This involved not just one-off lab experiments, but repeated testing over many years. I think in that study, as many as 30 years of large numbers of people. So the position when the Manchester Newcastle study was funded, obviously, what should we do? Obviously, time measurements were important, but we also needed to put in as much else as we could get away with, given the constraints of the time we had, about an hour a group to test people. There was no hope then in brain imaging, and that only occurred in the 2000s, right at the end of the study, when we could, on a subset of people in Manchester, obtain brain images. First issue, with longitudinal studies is what is the time course of rates of change in average performance. And that's how things look. You get a bumpy curve, and that's how things should be. So clearly God has screwed up again. The second issue was inter-individual variability. People age physically, but also neurophysiologically and mentally at markedly different rates. And the variability increases as you examine groups of different ages. So the older the group you examine, the greater tends to be the difference between the best and the least good in the group. And can we relate this to physiological and social factors? Can we say why some people are lucky and other people less so? A third issue is what factors predict survival. And we enrolled 6,000 Mancunians and Novocastrians, initially 55 to 92, and followed and documented all survivors over the next 30 years and recorded all deaths. We took 3 intelligence tests, the age 4, 1, and 2 in the Conceptual Culture Fair, and we examined crystallized intelligence because there was evidence that crystallized intelligence from John Horn had pointed out that crystallized intelligence learned things like vocabulary decline less rapidly with age than does fluid intelligence, which is usually a time-based measure. And we also had verbal memory, visual memory, and information processing speed, again, speed tests. And we included health and social factors, so depression, 2 different inventories, general health. We had some examined by physicians, but mostly it was personal details and self-report, income, and so socioeconomic advantage, and dates and causes of death were also recorded for those who died. That's nearly half the study, and before the study ended in 2004. So what are the approaches you can adopt when you've got this massive data? One is to track the change backward from significant events, and death is clearly a significant event. It's hard to identify and quantify all the things that happen to people on their lives. So a useful device is to count time not forward from birth, but backward from death. So this reveals what was called in the literature classically as a terminal drop, an unpleasant phrase in performance on nearly all mental tasks. And we had survivors and deceased from the Newcastle and Manchester study, and Mary Lunn and Danny Wong analyzed these. And that's what you get. The W things here are those who withdrew. D stands for deceased. So at this point, these are the starting points for people who withdrew and were deceased. These were people who died and withdrew and deceased here when they reached there. This is withdrew on the first session. They weren't very happy with the study, and they might not have been very well either. And so it goes. But generally speaking, the nearer you get to death, or the nearer you get to abandoning the study, which is a proxy for not feeling so good about the world in general, and perhaps yourself in particular, the less your cognitive performance as previously measured has been, and the greater the decline you show. So another thing to do is to look at what things contribute towards survival. And Stephen Akile wrote this very optimistically titled paper in 2016 based on the Manchester study. And here you see the rank order of the conditions which have effect. Being a woman is a very good thing because it lowers your mortality risk like anything. Subjective health, up in the top right-hand corner of the screen, my screen anyway, is also a marvelous thing. And you have to be careful how you measure it. This was measured by asking questions, but we asked 3 sorts of questions. The wrong sort of question is, how do you compare with other people of your own age? And that gives you rather poor answers, because, as you might expect, because a lot of people who would have been your age are dead already, or who are in a worse plight than you, and so you feel pretty good about being alive. But if you're asked, how much have you changed during the last 5 years, then this is very informative, and it's a very good index of mortality. The others I won't go through in detail, but it just shows you what you can do, and it also shows you we don't need convincing already that smoking is a bad thing, and that processing speed, being fast, is a good thing. That and other analyses also show effects. Socioeconomic disadvantage, education, that goes, of course, very strongly with socioeconomic disadvantage rather than disadvantage. Illnesses we've dealt with already, loss of mobility, which accompanies illnesses. And the psychological measures which matter are depression. The more depressed you are, the poorer your outlook. Intelligence, a decline in intelligence, and low intelligence has poor outlook. And here we get information processing speed, the Burton variable, if you like, which is a very powerful index. So decline in intelligence causes a risk factor, even after all these other influences are taken into account. So that those who show declines of 10% or more over 10 years had 2 times, and those who showed declines of 20% or more had a 2.5 times increased risk of death. Participants who scored were constant over 4 years had no greater risk of dying than those who scored were increased by up to 20% by practice of the tests. So predictions for dropout were similar. These are statistics by Mary Lunn, and they bring up this idea of PrAC for the first time. More of that later. This is just to show you that you can, and we did, break down deaths by cause, and you get the usual things. You get heart disease and cancer have large effects. Combinations of these things have large effects too. What might be a surprise to some is that accidents, dying of an accident, is also a large effect, but you must bear in mind that most of the accidents suffered by older people, which result in death, are due to falls or acts of clumsiness, which are due to motor incapability, which are due to being in a pretty bad way generally. So to check that current gross brain measures predict previous rates of cognitive change, we've taken death as a gross brain measure, a dead brain. But if you take measures from live brains, which we were able to do in the 20th year of the study, and age-associated brain volume is one I concentrate on here, but it's not the most important. So you can track back from that, as we did from death, and you find much the same thing, that if you have high levels of brain shrinkage measured towards the end of the study, then these predict very well the rate over which you've declined during your previous years of testing, which could be up to 20, depending on your year of recruitment. And this is with Saeed Ibrahim, Mary Lunn, and Margaret Scott, and Neil Tacker, and others. But we mentioned practice, and this has been the elephant in the room. It's been very widely neglected, and many, perhaps most published studies have ignored it. Watershy and Paul Baltus in the 1960s warned that during a study, when people were given the same tests again and again and again, they were likely to grow better at them. But this was very widely ignored because there was nothing to do about it at all. There were no statistics which could cope with it, and though Shy and Baltus identified the problem, they didn't suggest a solution. Once the solution had been found by Peter Diggle, it was very difficult to get our colleagues to accept this. And the first referees accused him of voodoo statistics. But it was a new invention in statistics, a new way to analyze and take amounts of practice effect into consideration. But fortunately, his statistics were available when pathologies were examined, and Mary Lunn and Danny Wong were able to incorporate them. And this showed some interesting things too, because if you looked at the rate at which people learned during tasks, the rate at which they improved declined the nearer they were to depth and the worse their health. So this is the rate of improvement with practice on tasks during a longitudinal study is itself a very good marker for brain changes which are associated with, with physiological changes, which produce, of course, neurophysiological changes. Further than that, there's some optimism because, like all people launching longitudinal studies, when you wrote our original proposal, we drew a blanket over the fact that practice effects might occur because we'd been asked how we could cope with them. So we thought that we'd get away with it, as everybody thought they'd get away with it, by using very simple tasks, so simple that one thought that they wouldn't improve with practice, and administering these tasks at 4-year intervals. So you'd assume that a simple task, like a reaction time task, over a 4-year interval would show no improvement. You've guessed from what I've said already that you did. And indeed, we were able to take people who'd missed testing sessions, and so over intervals of 8 or even 10 years, they had, that is, intervals of 8 or 10 years between successive testings on very simple tasks like intelligence tests and memory tests and reaction time tests, they did show improvement. Though, as I will remind you, this improvement would be much less in people who were either nearing death or were suffering from known pathologies. So then at last we were able, at the end of the study in 2000, we were able to look at the gross age-related changes in brain. Gross because image analysis is very intensive, and we could only at that time muster for the money we've got groups of about 150 people. So we obtained and used gross measurements, that is brain blood flow, gross brain volume, that's the difference between current skull volume, which we assumed would not change, and current brain volume, which would be less in an aged brain, and white matter lesion proliferation. And a paper with Dan Lung and Ometse Mokape showed brain blood supply was the key to the other brain changes. So as brain blood supply changed, gross brain changes, specifically white matter intensities and overall brain shrinkage occurred. And declines in mental abilities in this study, specifically in decision speed, which turns out to be the most sensitive marker for brain changes, decline with brain changes. So you've got a causal sequence here. Blood supply to the brain keeps things going, and this accounts for perhaps the widest positive news in aging studies, which is that if you want to keep going into old age, you do as much exercise as you possibly can and keep your brain blood supply adequate. So how do you manage this analytically? It pays to look backwards from current status to rates of previous changes. Now, the thing that is mostly associated with our sensitive index, remember, reaction times, and indeed also fluid intelligence, is white matter lesion intensity. And there are huge Dutch studies, mainly carried out in Rotterdam, and I've listed these for you. And I won't bother you with brain interpretation because I can't, but I'll show the pictures as a matter of pride. So measurement of overall white matter hyperintensities in the entire brain are more strongly associated with speed than with other measures. And the effects are stronger for white matter abundance in some areas, i.e. medial temporal areas, than in others. And overall white matter abundance also affects other mental abilities, notably verbal memory. So white matter in abundance is a key measure. It accounts for its progress accounts for the decline, the amounts and the rate of decline and other measures, and particularly in information processing speed, and also of interest to all of us, of course, in intelligence too. And white matter intensities are to some extent selective because they affect performance on different tasks depending on which brain area they occur. This hadn't been the case, of course, the data would have looked extremely suspicious because localization is the key idea in brain image studies. And here you get changes in verbal memory and age in ears with at MRI and cardiovascular symptoms, so that with white matter intensities are associated with cardiovascular symptoms and with age in ears, and changes in verbal memory decline as white matter intensities increase. So that the strength of the associations is really remarkable because white matter lesion abundance accounts for nearly all of age-related changes in GF and speed. And that is for about 98% or more of the proportion of variance in speed that is associated with differences in people's ages. So 98% of the age-related changes in speed are accounted for by measures of white matter abundance. And of course, looking backwards, white matter abundance at a particular time, paper with Saeed Ibrahim and Mary Lunn account for the rate of change previously. So brain blood flow and gross brain sinkage are combined. Effects of intelligence are almost entirely mediated by the effects on processing speed. That is, if you enter both gross brain shrinkage and white matter lesion intensities and brain blood flow into your analyses, then you can account for declines in intelligence and in processing speed, but the decline in intelligence is almost entirely accounted for by processing speed. It's entirely related to the effect of processing speed. At any age, processing speed and intelligence are strongly correlated. Particular interest that after differences in processing speed are taken into account, the effect of age on intelligence are almost completely removed. And the proportions on the different intelligence tests we used are quite similar. So it's between 89 and 99 for the Cattell, 80 to 96 of the AH4, and 98 to 100% of AH4-2. Right, and so we at last reach the summary. As we grow old, our health declines, pathologies increase, brain blood supply reduces, and death becomes more likely. Pathologies reduce brain blood supply and so global brain efficiency. These effects cause declines in mental abilities. Declines in mental abilities are good markers, that's not direct causes of progress towards mortality, but of rates of further declines. Most sensitive cognitive markers that we could find are declines, not overall lifetime levels of intelligence or of decision speed. This is worth stressing because if you're slow, either in the sense of being not very bright or of having slow reaction times, this is not a counterindication of itself. It is a change in these measurements which matters, not their level, lifetime level. So differences in speed predict between 80 and 100% of that proportion of the differences in intelligence between individuals that are associated with differences in their ages. And differences in speed are strongly associated with gross age-related changes in the brain. For example, best example is white matter hyperintensities that seem to be driven by age-related declines in cerebral blood flow, which is, to reassure you, increased by exercise, which incidentally I never take.
Very good. I believe again that Pat Rabbitt is not with us today, so we won't have a question period, which means we should probably give it 5 minutes so that the final talk begins at the expected time. We'll see you all again in 5 minutes. Hi, Jennifer. Thanks for joining us. I believe we're live, so we should conduct ourselves accordingly. So we'll just begin your talk. It's recorded, isn't it? So it's appropriate time, and then you can join us for questions afterwards.
Brilliant, thank you. John, will I just give a couple of minutes? Oh, Pat is here.
Hello.
Hello.
Unmute your speaker or increase volume.
Yeah, we can hear you fine.
Oh, yes. Sorry about this. It was a transition between computers.
Yeah, well, I'm very sorry. I did see an attendee called Patrick, and I thought, what's the chance that that's Pat? And decided there was no, I didn't know how to find out. Actually, I now realise I could have found out by sending you a private chat, so that was silly of me. Anyway, I'm sorry. I think we've probably discouraged any questions, unless some suddenly pop up, but I think that would be a surprise at this juncture. Anyway, my deepest apologies. Yeah, I haven't seen anything pop up, I'm afraid. So.
I'm sorry. I'm obviously representing some age-associated effects on use of technology.
I think I'm at least as guilty as you are. You just ended up in a slightly unexpected place, but
right. If there's a question, I'll try, but can't see anything.
No, I don't see anything.
There is a question in the Q&A from Frank.
Q&A. Francesca Happe. Wonderful talk, Pat. Can you say why being a woman is so good for aging, please?
No idea. No, there are a number of explanations, but I wouldn't pick any of them as being particularly credible at this time. But there's sort of sociobiological explanations have been attempted, like grandmothers are useful to rear a next generation, but grandfathers aren't. That sort of explanation, but all unconvincing.
Well, since you're here, Pat, I'm going to ask you a question myself. So what's your thinking now, after so many years thinking about it, on how the overall message seems to be a very global one, not totally, but very, very global? Is that because you think there really is one global process, or is it because there are many things that are also correlated with one another and with, you know, generally getting unhealthy and older, that it never is possible to pick them apart?
Well, it is possible, but it would require very large numbers, because I think with different pathologies and different interruptions of blood flow, you'd get different areas of local damage in the brain, and so different patterns of local damage. And to look at the effects of all of these individually, you would have to have huge numbers of people all scanned and all studied for long periods of time. I think I can only offer gross data because that's all that was available to us. It's all we could collect.
Okay, thank you. Well,
thank you.
Extremely interesting, and thanks very much for joining us.
Oh, great pleasure. Thank you. Thanks for the invitation. Thanks for having me, as people say on television these days.
Good. Well, till next time.
Yep.
I think it's time for us now to go on with the last talk in the symposium from Jennifer Cook. Again, it's pre-recorded, and then Jennifer will join us for questions at the end.
Doesn't hear anymore.
Hi, this is Jennifer Cook from the University of Birmingham, and I'd like to start my talk today by saying happy 75th birthday EPS. I've had a lot of help and support from the EPS over the years and have benefited greatly from the multiple meetings that they have every year. And so it's really nice today to have this opportunity to say a big, big thank you and a big, big happy birthday. So my gift to you, EPS, is this talk entitled Flexible Mechanisms for Social and Individual Learning. So social learning, what is it? Well, social learning concerns learning from, observation of, or interaction with others. And this stands in contrast to what we might refer to as individual learning, or learning via trial and error, or figuring things out for yourself. Now, social learning is a topic that has been of interest to academics from a wide variety of different fields. Psychology, of course, but also ecology, evolutionary biology, and even philosophy. And this is because social learning is thought to be of key importance in cumulative cultural evolution. The argument is that by learning from other people, we can avoid having to reinvent the wheel, and thus each new generation can make considerable advances on the achievements of previous generations. Now, in the social learning literature, there has long been what I think is a really interesting debate between two different schools of thought. Theorists in the domain-specific camp argue that living in social groups has favoured the evolution of social learning, and thus there are special mechanisms for social learning that are adaptations for dealing with the pressures of group living. So in other words, at some point in evolutionary history, we had to develop special mechanisms for social learning that are distinct from those mechanisms that we use for learning from our own individual experience. Domain general theorists, on the other hand, argue that all learning, including social learning, can be driven by the operation of a few general learning principles. So in other words, it doesn't matter whether we're talking about individual learning or social learning, but we're still using the same general mechanisms. For example, principles of associative learning or instrumental conditioning. When cognitive neuroscientists have looked in the brain at the mechanisms supporting social and individual learning, some of them have found evidence consistent with the domain general view. So in other words, some studies have found that the same brain areas and the same generally dopamine-mediated computations are involved in both social and individual learning. However, it's also the case that some studies have found dissociable neural pathways for social and individual learning, and other types of dissociations between these two types of learning. These studies are consistent with the domain-specific view because they find special pathways or special mechanisms for specifically social learning. So regarding the question of whether there are special mechanisms for social learning, evidence from cognitive neuroscience is mixed. In this talk, what I'd like to start with is to zoom into some of these dissociations between social and individual learning. And I'm even going to show you some examples of dissociations from my own lab. In the second part of the talk, however, I'm going to look at some alternative explanations. In particular, I will argue that in most studies that have dissociated social and individual learning, the individual source of information is primary and the social source is secondary. And therefore, I'm going to argue that dissociations between individual and social might be dissociations between primary and secondary. And finally, I'm going to try to show how this idea actually helps us to resolve conflicting findings in the neuroscience literature. To help me to give you some examples of dissociations between social and individual learning, I need to first explain a classic task that is often used in this literature. This is a task that was initially developed by Tim Behrens and Matthew Rushworth and other colleagues at the University of Oxford, and has been adapted in various ways and used by different groups around the world. On each trial in this task, participants are required to make a choice between a blue and a green box in order to win some points. Before they make their choice, they see a thin red frame representing social information. In some experiments by some lab groups, this represents information from a confederate. In others, including ours, it represents the most popular choice selected by a group of participants who previously played the task. Participants know that the correct answer will flip between blue and green, and that the social information will flip between phases where it's very useful and phases where it's not very useful. Thus, participants know that they must use their individual experience of reward outcomes to learn from trial to trial about the blue and the green boxes, and they must use the feedback to learn from trial to trial about the veracity of the social information. In other words, participants must engage in both individual and social learning. A number of years ago, we used this task to investigate the way in which personality traits linked to popularity and leadership relate to social and individual learning. We found that socially dominant people, who naturally tend to emerge as popular social leaders in the workplace, are more optimal in their social learning compared to people who are low in social dominance. And, most interestingly, I think, we showed that social dominance was a significantly better predictor of social compared to individual learning. In other words, we showed that social dominance dissociates social from individual learning. More recently, in a double-blind placebo-controlled study, we showed that the dopamine and noradrenaline transporter blocker, methylphenidate, impacted on individual learning, but not on social learning. More specifically, when participants had taken this dopaminergic drug, they were better able to adapt to the volatility of the individual information, those blue and green boxes. Meaning that when they were in a relatively stable phase, for example, when the blue box was rewarded for a long period of time, they had a low learning rate. And then when they were in a very changeable, volatile period, where the correct answer was flipping between blue and green, their learning rate shot up. And we found that this was true both when we fitted a Rescorla-Wagner style learning model to the data, and also when we ran simpler win-stay-lose shift type analyses. Importantly, however, the drug, methylphenidate, did not help participants learn about the volatility of the social information. So far, I have shown you two examples of dissociations between social and individual learning. Social dominance correlates with social but not individual learning, and methylphenidate affects individual but not social learning. So here we have patterns of data that are consistent with the idea that there are separate mechanisms for social and individual learning. And if I had longer to speak about this, I could show you similar examples from lab groups around the world. In this next part of the talk, however, what I would really like to do is to explore an alternative explanation. And I would like to argue that in tasks like the one that I've shown you, the individual source is primary and the social source is secondary. And thus dissociations may comprise dissociations between learning from primary and secondary, not social and individual sources. So why do I think that the individual information is primary? Well, I think this is the case because in this task, these boxes, the blue and the green boxes, which comprise the individual information, appear first. They are highly salient, big colourful boxes as opposed to this thin red frame. And also, the outcome information comes in the form of a small blue or green box, which primarily informs participants about whether the blue or green box was correct on that particular trial. And then from this information, they can additionally infer whether the red frame therefore surrounds the correct or incorrect answer. So to summarize, the boxes comprise the primary source because they appear first, they're highly salient, and they're in the same frame of reference as the outcome information. Now, going back to the methylphenidate study that I showed you previously, the pattern of results that I showed you could be a dissociation between individual and social learning, but it could also be a dissociation between learning from a primary source, the boxes, and from a secondary source, the thin red frame. Now, we reasoned that if we had a control task in which participants learned from a primary source and a secondary source, and neither of them were social in nature, and if we didn't see this same methylphenidate-driven dissociation of the control task, then we could be more confident that this pattern that you're looking at now represents a distinction between individual and social, not just primary and secondary. So we recruited a second group of participants, and these participants completed the same task. However, we told these people that the secondary sources of information, the red frame, represented advice from a rigged system of roulette wheels, which sometimes produced helpful recommendations and sometimes produced recommendations that were not so helpful. So again, we told them that things were going to happen in phases to replicate the social version of the task. Here on this figure, I'm plotting the data from still the people that had the social cover story, but I'm just plotting this in a slightly different way now. So what I've done is to take the difference between the methylphenidate and placebo conditions. So now data points above the black dashed line represent a drug-induced boost to learning, and data points below the line represent a drug-induced impairment to learning. And just to remind you, in this group that thought the red frame showed social information, we saw that methylphenidate boosted individual, but it didn't boost social learning. We hoped that the roulette group would actually not show the same pattern of data, because if we saw that, then this would provide us with evidence that methylphenidate, it doesn't just dissociate any primary and secondary source of learning, but rather it specifically dissociates individual and social learning. However, as you can see from this figure, we see that the roulette group showed the same pattern. So what we saw was that methylphenidate modulated individual learning from the primary source, but it did not modulate learning from the red frame, the secondary source. There was no significant interaction between drug learning source and the social versus roulette cover story. And furthermore, Bayesian statistics provided moderate support for the conclusion that the effect of the drug was the same for the social and the roulette groups. So by including this control task, we've shown that methylphenidate dissociates learning from primary and secondary sources of information. Now, going back to our central debate, the data from our control task forces us to question the domain-specific view. This is because many studies that support this view have used a similar paradigm to the one that we've used, but without the control task. And therefore, they could be showing dissociations, not between individual and social learning, but rather between primary and secondary learning. So our data make us question the domain-specific view. But on the other hand, they don't actually provide positive evidence in favour of the domain general view. This is because in the experiment that I just presented to you, we didn't actually see any effects of the drug on social learning at all. So we couldn't actually show that social and individual learning rely on common mechanisms. Interestingly, however, it follows from our work that if we could make the social source the primary source, then we should see an effect of dopaminergic drugs on social learning, just like we do when the individual source is primary. In other words, if the domain general view is correct, both social and individual learning should be modulated by dopaminergic drugs when they are the primary source. To test this hypothesis, we developed a version of the social learning task in which the social information comprises a primary learning source. In this version, participants first saw social information, which appeared on the left or the right of the screen and comprised a highly salient big red box representing a majority choice of a social group. Participants had to decide whether they would go with or go against the social group. Participants then saw a thin green and a thin blue frame surround either the red box or not. And just like in the classic version of the task, participants were told that things happen in phases, such that sometimes they would be in a phase where going with the social information would lead to points, and other times they would be in a phase where going against the social information was correct. And similarly, there were phases where the blue frame indicated the correct answer and phases where the green frame indicated the correct answer. After making their final selection to go with social information or not, they saw a tick or a cross, which primarily informed participants about whether going with the social information was correct. And from this, they could additionally infer whether the blue or the green frame was therefore correct. So in this version of the task, the social information is primary because it appears first on the screen, it is highly salient, and it is in the same frame of reference as the outcome information. To test our hypothesis, we recruited two groups of participants. One group played this new version of the task and another played the old version, where the individual information is the primary source. And in a double-blind placebo-controlled design, we used haloperidol, which is a dopamine D2 receptor antagonist, to perturb dopamine system functioning. To remind you, we hypothesized that haloperidol would perturb learning from primary sources, but not secondary sources. Indeed, we found that haloperidol perturbed learning from the primary source and not the secondary source. In other words, there was a significant drug by information source interaction, such that relative to placebo, haloperidol reduced the rate of learning from the primary source. Numerically, you can see in the figure that it also increased the rate of learning from the secondary source, but this was not a statistically significant effect. Most importantly, however, haloperidol affected learning from the primary source, irrespective of its social or individual nature. In other words, it didn't matter whether participants had played the new version of the task, where the social information is primary, or the old version of the task, where the individual information is primary. We always saw the effects of haloperidol on the primary and not on the secondary source of learning. Indeed, Bayesian statistics provided moderate to strong evidence in favor of the hypothesis that there was no drug by information by group interaction, where group denotes which version of the task participants have played. So, to summarize all of this information that I've given you in this short talk, I first presented some dissociations between social and individual learning. However, I then argued that these dissociations may represent differences between primary and secondary, not social and individual learning. And finally, I presented evidence that social and individual learning rely on common dopaminergic mechanisms when they are the primary source of learning. So to zoom back to our central debate, the final study that I presented comprises positive evidence to support the domain general view that both social and individual learning share common mechanisms. Furthermore, realizing the importance of the primary versus secondary status of a learning source may also help us to resolve conflict in the literature. And I believe this to be the case because in studies that have shown that social learning relies upon the dopaminergic mechanisms that typically underpin individual learning, social source has generally been the primary and in many cases the only source of information from which participants must learn. But in studies that have found different neural pathways for social and individual learning, social information is generally comprised a secondary additional source. We suggest that these neural pathways are actually not specialized for specifically social and individual learning, but rather they can process both social and non-social cues and can flexibly switch between the two depending on which cue is primarily relevant for the task at hand. So, thank you very much for your attention. I hope you enjoyed this talk, and I'd like to finish by saying thank you to my lab group and to all of our many collaborators. Particular thanks go to Celia Hayes, Hanneke den Ouden, and Roshan Cools, who played major roles in the development of these ideas and the first 3 experiments that I showed. The final experiment was led by Alicia Rabitsky with help from Bianca Schuster and Sophie Sowden, so huge thanks to them for all their hard work on this experiment. We are close to uploading a preprint of the final experiment, so if you're interested in that, then check out our website or tweet me or email me if you want me to send you a copy. And a final vote of thanks goes to all our many funders, without whom we wouldn't be able to do any of this work. And this includes the EPS. So thank you very much, EPS.
And thank you. So once more, and finally, you can either type questions into the Q&A or raise your hand. While people deliberate, perhaps I shall start it off. Do you think that you might get different effects if you used a social learning context that was more related to what you might think was the ecology of social learning, for example, learning from older siblings or something of that sort?
Do you mean with explicit social stimuli, or just in that context of learning from other individuals?
Either one, I suppose.
So I think yeah, there's sort of 2 questions there. One is, if you used really explicitly social stimuli like videos of other people, would you see the same results? And I think that the answer to that is that I would predict that you would see the same results. And the reason I think that is because there are a number of studies that, instead of just using this simple red frame, they've actually used a video of somebody who's kind of pointing at the blue box or pointing at the green box, or sometimes they have some eyes that go towards the blue box or some eyes that go towards the green box. So they're very kind of overtly social stimuli. And in the studies that have used these overtly social stimuli, they have also given participants the probabilities about the blue and the green boxes. So participants don't have to do any individual learning, they just have to do the social learning. So in this context, the social learning is the primary source of information. And what they see is activity in the dopaminergic midbrain, and they see variation in social learning that co-varies with polymorphisms to the genes that affect the dopamine system. So I think that even if you use these really overtly social stimuli, you would still see this modulation of social learning by modulation of the dopamine system if the social source is the primary source. What you were referring to is what's known in the cumulative cultural evolution literature as a social learning strategy, which is where you select who you're going to learn from. So you were saying, like, maybe you would learn selectively from older siblings, or it's common that people learn selectively from experts or from the majority of the group. And in that's kind of like a sub part of the social learning literature, this social learning strategy literature. And in that literature, there's also the same debate going on. These social learning strategies, are they in our genes, or are they something that we develop through our experience using our domain general learning mechanisms? And there, Celia Hayes and John Pearce have a really nice paper where they've gone through a lot of these social learning strategies, and they've explained how they could come about via associative learning mechanisms. So I would predict that even if we did do a strategy-based task, we would still see the same results.
Thank you. We have a question in the Q&A from G. Kranaki. Thank you, excellent talk. I was wondering, can these findings have implications for social learning in individuals with autism spectrum disorders?
Yeah, that's a really good question, because a lot of my research is autism research, and that was one of my motivations for getting into the social learning literature, because there are theories that maybe some of the social differences that we see in adults with autism come about because something different has happened in the process of social learning throughout that individual's life. And if that were the case, and this were to lead to selective differences in social abilities, then the argument is that maybe they have selective differences specifically in the processes that underpin social learning. But if that's to be the case, then there have to be special mechanisms that underpin social learning specifically. And I haven't found any special mechanisms that specifically underpin social learning. So it suggests that if there are differences in the learning process in autism, these are probably more domain general differences. So that it immediately makes you think of the work of, for example, Rebecca Lawson, where she shows differences in learning about the volatility of the environment, which is a domain general difference that would have an impact on both learning from your own experience and also from social information.
Thank you. And I think time is more or less up. So I'd like again to thank Jennifer, fascinating talk, and all the participants today.